Hello to everyone, we are the Group 5, and in today's video, we'll be talking about DNA replication of eukaryotes. But first, what is DNA replication? DNA replication is the biological process by which a cell makes an identical copy of its DNA. This process occurs during the S phase of the cell cycle and ensures that each daughter cell receives a complete set of genetic material. Let us embark on this microscopic journey exploring the three phases of DNA replication: initiation, elongation, and termination. Here are the enzymes in DNA replication. DNA polymerase synthesizes new DNA strands by adding nucleotides and performs proofreading to correct errors. Single-strand binding proteins stabilize the single-stranded DNA, allowing for smooth replication. RNA primase synthesizes short RNA primers to provide starting points for DNA polymerase. DNA helicase unwinds the DNA double helix into single strands to allow replication. DNA ligase fill the gaps and repair the DNA strand created, joining Okazaki fragments on the lagging strand. The nuclease cleaves phosphodiester bonds between the nucleotides in DNA and RNA. And the topoisomerase relieves supercoiling tension in the DNA and prevents the DNA from getting tangled.
Hi, my name is Julie, and I will help you discuss about the start of the DNA replication, which is the first part, initiation. So, we have here the DNA. It looks like this. We know that it's a double-stranded helix structure. So, if we straighten out the DNA helix structure, it will look like this. Let us lay it down flat. So, we have here a pair of double strands, and we will look first at the adenine and thymine. Why? Because these two have two hydrogen bonds and they are the easiest to break apart. So our DNA replication process will start with the unzipping. So this is our origin recognition complex, in which a protein will bind into that complex. So this protein, it is called the helicase, and this is the one who will do the unbinding process or the unzipping process. It will be done through an ATP hydrolysis. So it will look like this now, a Y, which is what we call the replication fork. And when we have the replication fork, or when the unzipping comes, we have to secure it. We don't want it to be re-annulled, or we don't want it to be joined again. So we will now have these small proteins, the one that you're seeing right now, that small circles, they are the single-stranded binding proteins. Now we see here the primase going into the DNA. So this primase, they add up the primers. So the primers are the one which the DNA polymerase binds to, and then the sequencing will start. So that will be the start of the elongation process. And I'd like to tell you also that the small circle there, it is actually the topoisomerase. So they are the one who helps the DNA become uncoiled or to prevent supercoiling while the helicase is actually unzipping the strands.
During elongation, DNA polymerase takes center stage. This enzyme adds complementary nucleotides to the exposed DNA template strands, extending the primers and creating a new DNA. Base pairs are cytosine paired with guanine, while adenosine is for thymine. The leading strand is synthesized continuously, moving toward the replication fork. The RNA primase adds RNA primer, then DNA polymerase will continuously pair the nucleotides, creating a new strand of DNA. DNA polymerase only works in a 5' to 3' direction called the leading strand. However, the lagging strand runs in an opposite direction called the lagging strand. the RNA primase adds an RNA primer, then the DNA polymerase will add a short strand of pair in a 5' to 3' direction. The process will continue, and these pieces are the Okazaki fragments. The exonuclease will remove the RNAs, and DNA ligase joins them together, forming a seamless, continuous strand. Through precise coordination, the elongation phase ensures both strands are accurately copied.
The termination of DNA replication occurs when the two replication forks meets on the same stretch of DNA. The process begins with the DNA strands, here represented by the 4 black curved lines, moving towards the termination site. These red arrows indicates the movement of the DNA towards the central region. So when the DNA reach the termination site, it will encounter the termination factor. So this termination factor represented by these yellow circles. So these yellow circles or termination factors are proteins that will bind to a specific DNA sequence, causing the transcription machinery to stop. Then after the termination factors or the proteins bound to the DNA, the transcription machinery releases the DNA. Now the DNA strand is free from transcription complex. Now the termination is complete. The DNA strand will continue to move away from the termination site. The transcription process is now complete, so the DNA strand is fully terminated.
And thus, the cycle of life continues. DNA replication, with its intricate choreography, ensures that the blueprint of life is preserved and passed on generation after generation. This remarkable process is a testament to the precision and complexity of molecular biology.
And that wraps up the process of DNA replication of eukaryotes. We hope you learned something in this video. Again, we are the Group 5 and see you in the next video.